The mission of BendixLab is to understand how cells organize their surface, how the cell uses physics to organize the membrane proteins and the lipids, and also to understand how cells communicate with neighboring cells and with the environment using physical principles. And we also would like to understand how membrane curvature on the cell surface affects the distribution of proteins and lipids. Some project highlights from the BendixLab are, for example, filipodia, which are these small antennae that grow on basically every cell in our body. They are known to pull and exert forces on neighboring environment and on neighboring cells. But we recently found out that they actually have the ability to twist. So they can actually twist during retraction, and this was a very surprising finding.
We aim to understand how adhesion GPCRs respond to mechanical stress and how mechanical stress can activate this class of receptors. And the work involves combining molecular biology. For example, we engineer cell lines, we introduce functional mutations in the proteins of interest. Especially we employ optical tweezers, and that allows us to apply mechanical stress directly to manipulate the cells and the receptors.
So another project highlight is membrane repair. It's a collaboration that we have with cancer scientists. Because cancer cells, they are super efficient in repairing their cell membranes, their cell surface. We have investigated this mechanism together with cancer scientists by using our thermoplasmonic method to kind of cut small holes into the plasma membrane of living cells using these plasmonic nanoheaters. And while doing that, we monitor how annexin proteins are recruited to these holes and fixing the holes. Using the techniques of molecular cancer biologists, we can CRISPR edit or delete some proteins in the cell so that they get a deficient membrane repair.
My research methods that I use to study plant science revolve about advanced optical techniques, especially level-free imaging techniques, which they take advantage of backscattering of light in plants or in your biological sample. And therefore we don't need any genetical manipulation to be able to study the processes we are interested in. By removing the cell wall, we can use advanced optical manipulation techniques to manipulate the membrane or manipulate the cytosolic compartments of a plant cell, and therefore we can actually study mechanics of plant cells in more detail. So we use thermoplasmonics to manipulate the cell protoplast, and by using thermoplasmonic we're able to selectively fuse two different cell membranes and creating a hybrid cell. This new experimental technique is quite useful when you want to study responses of membranes, proteins or membrane material.
So in our collaborative project with BendixLab, we are focusing on understanding how the specialized structures called basal body give rise to cilia formation. And the cilia are beating in a synchronized way, and by doing so they generate fluid flow. This is extremely important, for example, in our airways to, for example, move the mucus along the epithelium. During the formation of the cilia, during formation of the multiciliated cells, these basal bodies, they must get distributed and patterned. And this is extremely important because without the proper distribution and proper alignment, the ciliary flow is impaired.
In my project, we try to quantify the force required to the force cells through receptor beat bonds. Specifically, we do so to correlate these force measurements with signaling activity of the receptors. In parallel, we also work with single molecule studies aimed at understanding how the receptor interacts with ligands, and we do so in supported lipid bilayers.
We developed a platform to investigate the mechanical activation of adhesion GPCRs in living cells. We can use the optical tweezers to control a bead very specifically and assert a force on the receptor directly in living cells. We can do it in different directions. We can pull or we can push. And as we do that, we have inside the cell, we have a fluorescent marker for activity. It is in the cytosol and it goes only to the membrane if the receptor is activated.
My project involves mechanical perturbations of different cell lines, and that can be either via stretching or compression or optical manipulation with optical tweezers.
My research focuses on the crucial questions how DNA damage is repaired. This is absolutely crucial for preventing cancer because every cell experiences about 10,000 DNA lesions each day, and accurate repair is absolutely necessary.
So another focus of our lab is to investigate applications of thermoplasmonics. So it can be used for manipulating cells and manipulating the cell membranes, but it can be used for a number of other purposes also. For example, you can use these nanoheaters as a cancer therapy agent. You can inject it into humans or into mice and then look at how these particles or nanoheaters accumulate inside cancer tumors. So this is called a photothermal therapy, which is a very promising therapy for future applications.